cancer cell biology, and what we are going to see is essentially, uh, as you know, that it's a huge thing in the way that if we just think philosophically, that uh, in IH it takes a little bit of time, but NIH and CDC, they together actually do many of these studies, and particularly CDC actually publish an yearly report. It takes about almost a year or two to come up. Like I think the latest now is available 21, not even 22. That which are the top causes for death in the United States, and I mean like for all reasons. Like even they include like a car accidents, automobile, or anything like that. But what we just wanted to say is like a cancer was at number two, and heart disease was actually number one. But cancer actually pretty much almost at the same, or it just passing the heart disease. And it is predicted that in the next couple of years, it will be the leading cause of death in the United States and pretty much all developed countries, but not developing countries. So, and there is a reason for that. Is that are we doing something wrong? No, we are not. But the thing is that what happens that due to the advancement of medicine, the developed countries actually have much more life expectancy than the developing countries, where the life expectancy is significantly lower. So essentially, what happens, the more you live, your cells will divide more time, and chances of making mistakes is just a statistical number, and that will accumulate and can lead to the cancer. So these, all the degenerative disease will increase with the life expectancy, as we can see that shift. Like in 1940s, 50s, infectious disease used to be the leading cause of death. So with the antibiotics, antivirals, vaccine, and everything, that went away. And then these things came. Now, heart disease was one of the preopposite one with the food habits and all those kind of things. People are taking care of those. I mean, like, just think about that, McDonald's never used to, when I was like 20 years old, have you ever seen that you have a grilled chicken wrap in McDonald's? So people modify and do that. And these help in some of the cases, but cancer. So that's the a big picture of that. And so that's just, I just wanted to point out that that's the way to think about it. All right. So, and pretty much, you know, that why it is an important and a touchy subject, that no matter what, like every family, everyone, someone, their friends, families, family friends, or someone, they have touched with cancer. So, I mean, like it's a relevant, so you will be able to connect that. All right. So the first important slide is this, and pretty much everything in highlighted is a part of the question. So very straightforward type of question. We just ask, which are the following are characteristic of benign tumor, or which of the following are not characteristic of the benign tumor? And the same way the other side, malignant or metastatic, we can say is like which of the following are characteristics of the malignant or metastatic tumor, and which of the following is not a characteristic of malignant and metastatic tumor. So just to review, very straightforward, that this is as simple as, like, it could be a larger tumor, of course, but a simple skin tag is basically a benign tumor, which is an uncontrolled cell growth. And a polyp that is found in colonoscopy, which is benign, is an example of that as well. Sometimes it can get bigger, but most of the time, if they are small, they usually do not cause any problem. But they have accumulated some mutations in the cell that actually make the cell cycle throw off, and the cell cycle is not controlled. It is not too much bad, but it is, to some extent, it cannot control the cell cycle precisely as a normal cell, and it's dividing and localized. And then what happens that if there are bulky or biologically active, like a gland which become hypertrophied, like it become bigger, and then it's secreting hormone in a larger amount that you do not want. At that time, it could be a problem and may be removed. Other than that, usually they do not have any secondary side effect other than the bulk or biologically active substance production. Now, for malignant or metastatic, what happens that, as you know, that these cells will actually accumulate more mutations, and now what they do that they will not be attached to each other, and essentially this you can connect that you have the in between the cells you have the adherence junctions, you have the hemidesmosomes, you have the desmosomes, and these mutations will allow them not to have those linker proteins in between the cells and in between the ECM and the cells not expressed properly, so they do not remain localized in one tissue, and they will start moving out of the tissue and establish themselves somewhere else. And that is essentially the colonizing territories normally reserved for the cells. Are mutation just less in benign tumors? Yeah, I mean, like that is exactly correct. So basically what happened, Stephanie asked the question in the chat that we actually think that 3 mutations, minimal up to 7, and how these mutations actually work in function. Remember that one thing is so important, that why we do not get that many cancer or benign tumor as such, because all these mutations need to happen in a single cell, If these mutations are in 3 neighboring cells, all of them have these mutations in all combination, even the 7 mutations, that actually does not affect. It has to happen, all of them. So essentially, just think about this way, that one mutation is on a tumor suppressor gene that is not many active. So if that is not active, there is a secondary gene which will still stop the cell cycle or kill the cell, like a P53. Now you have a P53 mutation, it will not be able to stop uncontrolled growth. Then on top of that, you have apoptotic protein, like a caspase pathway protein, which is supposed to kill the cell, is damaged. So somehow these combinations will not allow a cell which has unscheduled DNA synthesis and growth, cell division, not stop, and it will just grow. So that's essentially the same thing. And essentially what we are seeing that, say for an example, just give a very crude example. Suppose in a cell there is like a one protein, like let's say for an example, a tumor suppressor protein and a protein which actually throws off this P53, ATR, ATM, one of them is mutated. So P53 is good to kill the cell to some extent, but not all of them. So you have a small tumor that is growing, but rest of all the cell ECM proteins and all the linker proteins in between, they are intact, so it cannot get out. And then on top of that, you have a couple of more mutations where you have no control of the growth, no on cell division, and then you gradually lose the cell-to-cell junctions, which actually keep them in the tissue, and that's exactly how it progress over the period of time. So the benigns, there's always a chance that it can get malignant. That's why whenever you see that, the doctors actually do the biopsy to just make sure that it is not. But this is basically a continuum as such. All right, so we already talked about this. So these are cells have a contact inhibition, but the cancerous or the neoplastic cells, which are transformed, they do not actually have any contact inhibition. So as you can see that normal and the transformed cells, they can actually pile up the transformed cells, and on the normal cells, they grow in a monolayer. If you have a Petri plate that you're growing the cells, they will spread, and after that, once they touch each other, they will essentially start dying. But it is not the case for the cancerous cell. All right, so next, these 2 slides are very important. You will have multiple questions out of these. So one of them, and this is basically what I called as a 360-degree coming around from cancer biology to the first chapter, cells. So let's take a look at each one of them, because this you need to understand. We will have a critical thinking question from here. So first, you have commonly observed in a transformed cell, you have a diminished adhesion to the surface, therefore able to maintain the rounded morphology. You can see that these are some of the cells which are still flat, how it should be, but these cells are rounded, essentially. These are scanning electron micrograph, essentially. And then failure of the actin filament to organize into stress fibers. See that right here, the shape of the cells is actually kept in check by proper polymerization of actin. And this is a failure for actin to do so. And thus does not mean that there is any actin in there. It is essentially most of the time what happens that how the actin filaments essentially do not do their function is the actin needs to get a cell signal to do so. So it just need a signal to keep it flat. And if that signal is absent, it will just do aberrant things that it's not supposed to do, or it will simply not do anything. But it is not the absence of actin. If there's an absence of actin, the cell will die. It's not. It's just signaling or regulation of the actin that is impaired. Then reduced external coat of fibronectin. So fibronectin is right here, is one of the ECM proteins. So you definitely underline that. And this is one of the ECM proteins, and this helps to keep them in the tissue. Now, this is also very interesting that the cell surface actually contain a lot of proteases, and there are functions because it actually processes different things. Say, for example, we all know for SARS-CoV-2, this ACE2 angiotensin converting enzyme, that essentially is the receptor. And we always constantly say that a protease, a furin-like protease, actually cleave these viruses, their spike protein, and then help them to bind. And that's absolutely normal distribution of many proteases are present, and they actually do different types of function, organization, receiving signal, processing molecules. But here there is an activator for these proteases, and this is called plasminogen. So if a cell get mutated and produce too much plasminogen, and it will cause too much extracellular protease synthesis, leading to extracellular proteolysis. So again, repeating, if the cell produce too much plasminogen activator, it will increase extracellular proteases, protein digesting enzymes, generic, and increased extracellular proteolysis. So what proteolysis will do, it will now start digesting essentially the anchoring proteins of your adherence junctions, desmosomes, hemidesmosomes. So they are all proteins, and if this proteolysis increases, including say fibronectin, which is a protein too. So this increased proteolysis will just digest all these proteins, which are the glue proteins, adhesion proteins, or essentially what we call this CAM, cellular adhesion molecules. This proteolysis will basically digest them, and that will lead to metastasis. They are not anchored in there. So we actually did a similar kind of experiment in the cell culture on the very first chapter. So what we said that when we have a petri dish of cells, like we have say right here, the previous chapter right here, we have these cells, not these, like this, and they are growing in a monolayer. And now it is like a confluent all over the Petri plate, and we need to passage this Petri plate of these cells, say 10% of that, into a new Petri plate so that they can grow. And that's how we just passage the cells. Now, to take these, and these cells adhere to the bottom of the Petri plate, essentially by secreting the ECM proteins. And if the cells by itself secrete, and these are usually, do not need that detail, but these are usually treated plastic. These are polystyrene, and the surface of them are actually etched. We cannot use glass Petri plates because these cells will not attach. So they are little etched, and on top of that, the cells secrete this extracellular matrix protein, and so which is essentially the glue, and keep them attached. So if we need to passage those cells from one Petri plate to other, we use a protease, either collagenase or the trypsin. Classic example of two proteases, collagenase and the trypsin. And also, we are going to see, we use your EDTA. And EDTA is essentially responsible for mopping out all the divalent or monovalent metal ions, including calcium. And now you know very well that your Ig-like CAMs or your integrin, they are all calcium binding proteins. And when you add your EDTA, that calcium is gone. And then you digest those remaining proteins, which are the cellular adhesion molecules by the collagenase trypsin. So they are partially digested, and the cells actually come out from the Petri plate. And now you can passage them. It doesn't hurt the cell. So essentially, after passaging, you will just wash them with the regular buffer. You will take the collagenase and the trypsin essentially out. You just expose them for a short period of time. And after that, you take them off. It will not digest the entire cell, or it doesn't have any effect. It just temporarily takes them off, essentially. So this is the analogy of the exact same thing. What naturally we are using collagenase, trypsin, EDTA to take the cells off the Petri plate, whereas in our natural body, just increasing plasminogen activator, reducing the fibronectin, as we are increasing the proteolysis, it is achieving the same cell as if from the tissue, the cells being lifted up, and now they can move in another area and become metastatic. So just know this connection. All right. So after that, we also do have the second commonly observed in which are growth and division abnormalities and unusually high density, lowered requirement of growth factors. Loss of anchorage dependence can grow even without attachment to solid surfaces, and loss of contact mediated inhibitions. We already talked about that. And importal can continue to proliferate indefinitely, can cause tumor when injected to a susceptible animal. Now here, I will underline the susceptible animal, this one, because what happens that if you just simply put any DNA, cancerous DNA or tumor derived cells into a healthy animal, most of the times, even in mouse or any subject, it will not always, sometimes it may do, but most of the time, the cells will not be able to create the cancer because it will be labeled as a cancer cell, and your immune system will actually kill them. And again, that happens in your own body as well. So you have to have some kind of mutation somewhere else where your immune system cannot keep up with the proliferation. So that's important. And this is also another important, the B, why you have lowered requirement for the growth factors. So essentially what happens that you have receptors for the growth factors. Many of them bind to the cell surface receptors or nuclear receptors, but sometimes they're in a mutation on those receptors and they are constitutively active. So even without growth factors, as soon as those faulty proteins are produced, we're going to see later, they are activating the entire pathway by itself. Receptor tyrosine kinase is a classic example of that. There's a mutation and receptor tyrosine kinase is produced and it dimerizes and it automatically will throw out the entire chain, and that will lead to uncontrolled growth, essentially. So these are some of the consideration that what you have in here, essentially. All right. So here, the word that I said that susceptible is an important word, and this actually shows that what susceptibility means. So you have a tumor, you can isolate the DNA from there, and if you just take some of these mouse fibroblasts, and then you transfect the DNA. So you use some of the lipid carriers, lipid nanoparticle. You just mix them in the cell culture. Those lipid nanoparticle will go into the cell. The cell will take up. I mean, like it just needs a few of them to take up. And if that DNA is actually transported into the nucleus and it has a homologous or heterologous recombination or mitotic recombination, rather a correct word. The mitotic recombination, it can integrate and you can have a transformed foci. So essentially what you are seeing that you have a cell and suddenly just a small foci of the cell, these are transformed and it just can arise. So you just only need that. So this is like all over the place. You have the normal cell and just a few of them. It takes a time. It's just not easy that it can be transformed. Now, you can just inject these transformed cells in these foci to a mouse, and it will develop the tumors. And this essentially is a classic experiment that it is a mutation in the DNA that is causing it. But to do this, you need either these type of mice. Either you need a SCID, which is a T cell, B cell deficient, which is the severe combined immunodeficiency. Those are called SCID. And the nude mice are only T cell deficient, not B cell. Now, the T cell is actually more important for killing the cancer cell because you have the cytotoxic T cell, which actually look for cancer cells more. B cell will also bind to some of the proteins which are abnormal proteins, and it considered it as a foreign protein. And if they're expressed on the cell surface, they will bind to it. But these are the ones which are the susceptible animals and essentially immunocompromised, have to be maintained in the laboratory. They do not have any fur. They are like this, just skin, and they can be used for this purpose as well, to study these. All right. So after that, we are looking at the general consideration about the proto-oncogene, tumor suppressor gene. Lots of highlighting. So essentially, we are just looking at the main two types. First, we are talking about the proto-oncogenes. And although we call oncogenes, which is cancer-causing genes, there is no genes that actually cause cancer. The purpose of that is entirely different. But essentially, what happens is, so essentially what happens is that these cells play a normal cell growth and proliferation. And these are positive regulators, essentially. And if you just think about that the cyclins are classic example of this, that you have the the CDKs, which is a cyclin-dependent kinases, they are present all throughout the cell cycle. But the cyclins are produced and degraded, and they actually activate the CDKs. And sometimes you have a mutation in the cyclin gene. They are constitutively produced, or they are produced under a transcription factor in a wrong place. And they just they're just constantly giving a positive input for the cell to go through the cell cycle. And that is your proto-oncogenes. And these oncogenes are the mutated version of the proto-oncogenes. And here, these are the gain of function. So essentially, think about this way, that the proto-oncogenes in a normal cell, when it is not dividing, the proto-oncogenes are not producing any protein. They are off all the time, normally. They are off all the time, normally. If the cell gets a proper signal to divide, only at that time, the protein will be produced, which means it's turned on. So that's the function. But what happens that when there is a mutation, gain of function means that the turning on switch is turned on constantly. It cannot be turned off. And that's the thing. And these are dominant genes. So essentially, if you just think about that there are two alleles, we have one pair of chromosome. So say one is capital A and a capital A. And both, so all it needs that out of those capital A and the capital A, suppose those are the two genes, and only one of the A's out of the two need to be mutated, and that will produce the protein. So that's essentially the idea. So if you just think about that if we just designate that both of the alleles in a chromosome, both alleles are essentially a pair of chromosomes are mutated, which is A and A. Very rare chance, doesn't happen. But if we say A and small a, that means the small a is good, that is not constitutively turned on. The capital A is mutated and is now constant on. And of course, that will lead to the cancer. And if they are small a and small a, that means their function of both the alleles are perfect. They are not producing any issues. And that's essentially. So I will just draw that just quickly, just what I said, that if you have a condition which is AA, this is essentially sufficient, and that's why we call it as a dominance. This is good, and this is good, but this really doesn't, and this is essentially bad. So you just only need one of the allele to be bad to cause this. On the other hand, if you just go to the other class of the tumor suppressor genes, is your normal function is inhibition of the cell growth proliferation. These are negative regulator. These genes are essentially on all the time. They are on all the time. Classic example is a retinoblastoma protein that we talked about, on all time, and these proto-oncogenes are essentially off normally. I'm just saying, so off normally. And if you turn it on, then the cell cycle goes on. A tumor suppressor normal function is inhibition of that. And here it is a loss of function. Here it is gain of function, contribute to the transformation of the cell. And this is a recessive. So essentially what is happening that if you have a condition where you have, suppose, just think about that it is A. And A is basically protein production. I'm just saying protein produced. Protein produced. Sorry, I'm just like I have a sticky mouse. Protein producing. And right here, if you produce the protein, that is bad because it is a positive regulator. Here, if both the alleles are producing the protein, this is good. If one allele is mutated right here, this is the bad. It's just the opposite one. This is the bad. And here, this was good, not bad. This was good. So here, this one is producing the protein. One is bad. But this protein is still producing tumor suppressor gene. So this is good as well. So there is absolutely no problem. So if you just only have that both of them A, that is, say, capital A is protein producing and small a is not producing, essentially. Protein not produced. Not produced. At that time only, it will essentially cause the cancer. And think about that, you have the two alleles of RB, and one RB is good, one is bad, no problem. But if both the alleles of the RB is this, then it can cancer, and that's why we call it as a recessive. So only homozygous condition, it can actually cause cancer, but this will cause cancer even in heterozygous condition. It just needs to produce that mutated protein. So know this very well, and this will do it. Any questions on that? You guys good? Good? Okay. Yeah, I mean, like, feel free to stop me at any time if I just need to re-clarify, or you can just write that right in the chat as well. All right, so these are essentially the thing that, remember that 3 to 7 independent genetic accidents required in the same cell. So why this number varies is just depends upon that what are the crucial mutations that is happening by accident in a cell. Sometimes more is required because if there is a mutation that can cause some problem in the cell cycle, but there are still mutations related, but still the cell cycle can be controlled by some of the proteins. But 3 means that very crucial pathway proteins, which are even looking on the first one. If the first one get mutated, suppose the second one is actually looking over the first one, and that is also mutated. Now the third one was looking after the second, and that was also mutated. And if they are very crucial in that pathway, that can lead to that. Whereas the 7, when we see this number is a little bit more, that means they are related, but they have a little bit less effect. So it just depends upon that. And as you can see, the age is directly proportional of none of these genetic to these genetic accidents or mutation, and increased probability of cancer as you age. And if you just go to the world life expectancy, you will see that essentially how it actually affects the developed countries essentially. So sometimes we can just give you as simple as a graph and just tell is say, for an example, like we just give you a kind of cancer. Suppose an incident of, say, colon cancer increase with age, and we give you a graph. And when the number of newly diagnosed cases in women in 1 year is plotted against the age of a diagnosis, how would you explain this data, essentially? So we just say that one cancer increased with age, and we give you a kind of graph. And the simple answer is that it takes the mutations in several critical genes to disable the normal mechanism of controlled growth. So essentially, over the period of time, the mutations are accumulating. When they are in these critical mutations, only at that time, there could be different mutations, but again, it was not apparent. And the chances of those critical mutation increases with age. And that is essentially the take home from that. So that will do it. And all right. and remember those numbers as well. So tumor appearance over the mice over expressing MYC and RAS oncogenes. I will talk about the experiment, but essentially the detail of the experiment is not that important. This just gives you an overall view of the additive effect of two oncogenes. One is a MYC, one is a RAS. And RAS, we talked about that. This is the RAS which essentially bind to the, when RTK, receptor tyrosine kinase, is activated, you have the adapter protein, which is a GRUB, essentially the GRB2, GRUB2, which essentially binds to the RAS, and that activates the entire kinase, RAF, MEK kinase, MAP kinase, or P90, RSK, and cell proliferation. And just a mutation is this valine at the 12th position can cause this. MYC is also a proto-oncogene controlled cell cycle. So what we are seeing that if you just only have a mutation in MYC in some of the mice, you can see that about 100 days of right here, 3 months plus, you start getting some of the cancers. RAS is a little bit more aggressive. You start getting cancer within 30, 40 days. But if you just see that MYC and RAS essentially just see that it just starts getting the cancer essentially within 30, 40 days. And see, by 150 days, 100% of them are dead, essentially. So how they actually do this experiment, and if you just reading the blurb, I just explaining that just in short, essentially. So what they do, that they have these MYC and the RAS, the mutated ones, under a promoter. And this is essentially called MMTV promoter. It's a murine mammary tumor virus promoter. So what happens that when they have these promoters under their control, it is tissue specific. So and this is like essentially in the breast. It's a mammary tumor virus. So this promoter actually goes into the and expressed in the mammary essentially after you make these transgenic mice. And then what they do, that they actually treat them with the hormone essentially to turn these, and these are essentially expressed by essentially if you just have increase their estrogen level, essentially those estrogens will turn on these MMTV promoters and drive the expression of the RAS. Means that this oncogene will be produced, oncogene is produced, and this is tissue specific. So they are going to express only in the breast or the mammary tissues. And that's how they actually can see that in the transgenic mouse, they purposefully mutated those, and they see these tumors. So it's written in the blurb, and I say it's like if you're just going through that, and if you just like how you do the experiment in this, and this shows the additive effect. You have just only one transgene, you have another transgene, just they're single. This is, you have a double transgene under this promoter, and you can just make this transgenic mouse and turn them on at your will by addition of the hormone in their diet. And that's how you can study those. All right. So after that, we are looking at, see right here. Yeah. So let's take a look at these essentially. Yeah. So after that, we are looking at the different classes of the oncogenes, and you have the proto-oncogenes. They are class 1 through 4. Tumor suppressor are 5, 6, 7, and some of them are kind of a borderline, like they are kind of doing both. And I'm going to explain that essentially. So first, what you have is some of the signaling molecule that can get mutated. We will just discuss not all of them, but just a fair amount of them. And then you have a signal receptor. We are going to see that the signal receptor, this is your class 2 proto-oncogenes, and this can get mutated. Then you have an intracellular transducer. So think about that. I'm just giving just one example, one of each. Not needed from here. You will have somewhere else. But this is a receptor tyrosine kinase, or GPCR. These are kind of a receptor that can get mutated. Intracellular transducers. This could be the G proteins, which is inside the cells, essentially GQ, which helps to activate some of the protein kinases, like a protein kinase C. On the other hand, this could be RAS, the RAS, RAF. These are intracellular transducer, mainly RAS, which we saw just the previous. This is actually a very, 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 very strong candidate for cancer. We see that mutation many times. That's your intracellular transducers. And then intracellular receptor, just like a signaling molecule, signal receptors can be on the plasma, sorry, not signaling molecule. Signal receptors can be on the plasma membrane, class 2, but if it's a nuclear receptor, that is also class 2. And then what you have, that you have the number 4 is essentially the transcription factors. And these transcription factors are essentially the product like a MAP kinase or P90RSK. These essentially translocate into the nucleus and bind to the DNA to drive those transcriptions. So essentially, if you have a positive regulation of those, then it will have a positive effect. So that's why they are proto-oncogenes. So these are all, if you are getting signal from these, it drive the cell cycle into division. So that's essentially the reason. On the other hand, if you just look at the tumor suppressor gene 5, 6, and 7, so right here is a cell cycle control protein. And cell cycle control protein, classic example, retinoblastoma protein. This is a essentially this is your RB. And then cell cycle control proteins also, along with that, you have these DNA repair proteins. So if there is a detection that there is a damage in the DNA, we know that P53 is one of the protein that is actually induced. We call the P53 as the guardian of the genome. That essentially call out all the DNA repair proteins. So P53 will be one of those examples as well. Now, if P53 can't actually stop the cell division yet, there are more mutations, then you have an apoptotic proteins, which are essentially the class 7. This will lead to the death of the cell. And as I said, is that if you're just think about this way, that you have a condition where you have, just like a hypothetical, you have a problem with your intracellular transducers. That means that they are always active, producing the transcription factor and going into the cells, into the cell cycle. So you have one mutation right here, and then then you have a problem with the cell cycle control protein and the DNA repair protein. So all these three can lead to the cancer. So now apoptotic protein still may work, but it's not sufficient. Or it may be intracellular transducers, transcription factor, cell cycle control protein, apoptotic proteins, DNA repair. You have 4 of the very strong mutations in those, and that can lead to. Now, happening this in a single cell, because these are all at the DNA level, these are all producing proteins happening in the single cell in all these crucial proteins, is a less chance. That's why we have these trillions of cells. They are dividing, but still we do not get cancer every day in every cell. And on top of that, you have your immune system to clear them out as well. All right. So let's, what's the time? 7:43. Okay. Is the internet still okay? Is it cutting or anything? No issues. So I think that the main thing that I'm still thinking, okay, all right, we'll still have to pass another 15, 20 minutes to see that if it replicates. But maybe the background is one of the issues. And I turned off all my Dropbox, Google Drive, everything which basically runs in the background. I turned all of them. So hopefully works. All right. So in continuation with this, so let's start with the module 2, which will essentially go into a little bit detail of the proto-oncogenes and again, gain of function. So let's take a look at each one of them. And each one of them is important for questions, not a large material. So definitely remember all of these, that each of these examples, what essentially happening, you can ask that this essentially will give an example of each one of them. In gene amplification, which of the following is happening? In chromosome rearrangement, which of the following is happening? So, I mean, like, we can just give you this and say it's like both of them are happening in here. So these are, but say chromosome rearrangement just does not mean constantly on hyperactive protein. That should be some kind of a mutation somewhere else. So let's take a look at one of them, that what is happening. So the first one is a deletion or point mutation in the coding sequence. This is like directly an amino acid is replaced by a different amino acid, and it just makes it constitutively on. So essentially that classic example of that will be the example of this RAS V12. Valine at the 12th position, if it is just turned off, converted, but in a different amino acid, it will be constitutively on, and the entire RAS receptor tyrosine kinase pathway will be on. So RAS will activate RAF, MEK, MAP kinase, P90, RSK, all throughout. So constitutively on hyperactive protein. This is one direct mutation. Then on the other hand, you can have a gene amplification. And how this actually happens, that we had in our gene, these are normal for maintenance of our genome, we have what is we call as a transposable elements. And these transposons essentially can cut a piece of DNA out from the DNA and insert it somewhere else. And they can duplicate the DNA in that process as well. The reason that is very interestingly, we actually have indigenous reverse transcriptase, which our DNA code for reverse transcriptase. And the question is that how do we even get that? That's an entirely different story. It's the virology class. I would love to discuss that. But essentially what happens that as the mammals and us, we are evolving, these retroviruses actually infected human beings thousands and thousands years ago. And these retroviruses essentially are integrated into the genome, and they get mutated. And they get mutated so that they cannot, those provirus, which are part of the genome, is not functional anymore. That means that they will not make the virus. Because say, think about this way, that somebody is having an HIV and have a gag pulp envelope protein, and that person will produce HIV if not taking drugs. And over the period of time, if that HIV is essentially integrated into the germline cells, that can be passed into the next generation. But what happens that what is a process we call is a fixation of these retroviral gene, that sometimes these are mutated, so it cannot produce all these envelope proteins, this protein, that protein, but maybe a functional reverse transcriptase remains in your gene. And these transposon and the reverse transcriptase has the ability to duplicate the gene. Why it is important? This has an immense importance in evolution, because what happens that sometimes if there is a gene responsible for adaptation to a new environment and it is fruitful, over the period of time, the duplicated of those genes have been selected. And that's why there are many genes which are duplicated so that they can produce more proteins for survival in the environment. So it has a positive effect. But unfortunately, what can happen that even in still today, in someone, those transposon and reverse transcriptase essentially can take a piece of DNA out, and then from there, it can just copy it and put it somewhere else in the genome. So you have, instead of one copy, you have multiple, like right here, 3 copies. And if all of them are under a strong promoter, they're all producing the protein. The protein is absolutely normal, but it is produced too much in amount. So think about this way, that you have a growth factor, like an epidermal growth factor. And you are just supposed to have I'm just saying, just empirically, completely, that you have epidermal growth factors produced from just only one. Now you have 3 genes producing three times amount of that growth factor. And that will essentially lead to enhanced cell division. Normal protein overexpressed, but it is not mutated, and it is just amplified gene, multiple copies, more protein. Then you have chromosome rearrangement. So the chromosome rearrangement can be 2 types. One, that this gene is intact, no mutation in the gene, and somehow this chromosome, these are essentially double-strand breaks, and then they swap these things, and these are essentially recombinations. And these recombinations, see that in the meiosis, in the germ cells, when you for the cell division, you have the crossing over. That is a controlled event. But you can have this mitotic recombination, which sometimes actually that happens constantly. But if there are sometimes at the same time two chromosome part actually break and then they swap and then they join those in a wrong place, you can have a promoter where there was a weak promoter. That means it is producing less amount of these gene product. Suppose in a normal condition, this promoter was red and it was producing just only one bar, one protein. One protein was producing. I'm just like empirically saying. Now, there is a mitotic recombination. It just received a very strong promoter. Now, this is a green promoter that is inserted instead of the red promoter upstream of that red gene. And now, instead of earlier producing just only one protein, now it's producing 3 times of the normal protein. So the effect of the gene amplification and this type of chromosome rearrangement is the same. You are producing too much of the protein, and that can essentially lead to the positive regulation of the cell cycle or driving the cells to go into the division. On the other hand, this is actually very unique, and we see in many different types of cancer, is the chromosome rearrangement can bring an entirely unrelated protein and just break a part of the DNA and insert it. And now it makes a chimeric, we call this a chimeric protein. And it's a constitutively on hyperactive fusion or chimeric protein. It is not a normal protein, absolutely abnormal protein, but it just constantly on. It is just constitutively active. If it is, we are going to see that a classic example of that. Suppose it's a receptor. It is produced in a different place, in a different way. And as soon as it is produced, it is active and signaling the cell to divide. So we are going to see that example of that, of the receptor tyrosine kinase, and we will see that. So these are the ones which are the different types of proto-oncogenes. And again, we can give you an example of that and just tell us that what is exactly happening in there. All right. So then we have the growth factor receptor. And again, this is the classic example of these receptor tyrosine kinases. We know that growth factor binds to these monomers and they dimerize, cross-phosphorylate, and then you have the whole pathway. In short, RAS, RAF, MEK, MAP kinase, and transcription factor and growth. Now, this is the one that I was talking about, and this is an example of this constitutively on hyperactive fusion protein. Chromosome rearrangement caused this, essentially. So what is happening right here? So this is a normal RTK. Sometimes it's called tyrosine kinase, TRK tyrosine kinase, or receptor tyrosine kinase. So these proteins, as you can see, that they are on the plasma membrane, transmembrane domain, extracellular domain, cytosolic domain. These are produced, and of course, you know this is a plasma membrane protein, so it got to be synthesized through the secretory pathway. That means as soon as the protein actually starts, it should have a signal sequence, either internal or it's an internal sequence. The ribosome will be brought onto the ER. It will just insert the protein onto the ER. It will go to Golgi, packaged for a plasma membrane, and it will go to the plasma membrane. That's essentially the signal. That's how it should be produced. On the other hand, this is a normal non-muscle tropomyosin. This is actually a cytosolic protein, essentially. Now, what happened that this is a chromosomal translocation, essentially, which is a rearrangement. This is your tropomyosin. And you can see this is the N-terminal, this is the C-terminal, N-terminal, C-terminal, and this is the RTK. And see that in the fusion, basically, it actually breaks off from here. So this RTK is not of this length. It is partial. And see that how it is essentially fused. This is the N-terminal of the tropomyosin. This is the C-terminal of the tropomyosin. Now, this entire extracellular part domain, almost all of it is gone from the receptor tyrosine kinase. So this is essentially the sideways of this picture. So we just put this two monomers of receptor tyrosine kinase sideways. So this is essentially the signal for the membrane and like transmembrane domain, essentially. And then you have the rest of the part. Now see that what happens. When normal receptor tyrosine kinase is produced, you have the N-terminal is in the exterior, And that means that it will be in the lumen. So, and that's, if you just remember, do not have to, but just remember. So it should have an N-terminal signal. It's a type 1 protein. So you have an N-terminal signal, it goes to the ER. That N-terminal signal is not there in the mRNA, and that's why, because it is already translocated. So it's not going to produce those, because now the N-terminal is tropomyosin, which is cytosolic. So when this protein is produced, it is not going to go to the ER because there is no SRP binding and taking them to the ER. The entire structure, the fusion protein, is going to remain in the cytosol. And you can see that there are 2 chains of the tropomyosin bound together, and that will be bound with the one molecule of the monomer of the receptor tyrosine kinase. And this fusion protein automatically dimerize the receptor tyrosine kinase. So automatic, as soon as it is produced, due to the affinity of the dimerization of the chimeric of the tropomyosin, it dimerizes, autophosphorylates. So this is become a cytosolic protein. Instead of that, it will immediately, if we just go to the previous one, so instead of that, this will do in the cytosol, it will activate RAS, RAF, MEP, MAP kinase, and the unregulated growth. So N-terminal extracellular domain, RTK, fused with this, and C-terminal is a So this is your new N-terminal. This is your truncated C-terminal of that, both protein. So this is essentially chimeric oncoprotein from the chromosomal translocation, and chromosomal translocation is essentially a type of chromosome rearrangement. So this is exactly what happened right here. So chromosomal translocation, a type of chromosomal rearrangement. This is under this. All right. So after that, we are just looking at just a couple of other examples, activating mutations, overexpression of growth factor receptor. This is a HER2 receptor. This is a EGF-like receptor, receptor tyrosine kinase, and just a single valine to glutamine essentially is an oncogenic mutation. And you can see that right here. This is what is called a new oncoprotein. The reason for this, that how this happens, that when these were valine, they have a kind of a conformation which doesn't allow the 2 monomers in the plasma membrane to interact. Only when it binds to a growth factor, that changes the conformation and bring them together. But if the valine is changed to glutamine, they, without any growth factor, they will automatically have affinity to each other due to the changed conformation of that, and they will dimerize, and they will just signaling for cell cycle. This is called a new oncoprotein, and this is a HER2 receptor for that. And this is an epidermal growth factor receptor, just like an EGF receptor. Remember that these are all receptor tyrosine kinase like receptor, but so remember that receptor tyrosine kinase is a generic name. There are hundreds of the families of there, and HER2 is one of them in the breast cancer. EGF, epidermal growth factor, is an example of that. Here you have a ERB oncoprotein, and this is a deletion. So see that this protein still does have a signal, which is a membrane-bound signal, and see that right here, just this membrane retention signal brings it to the essentially to the membrane, and it just automatically dimerized, and there is no external external N-terminal, and is constantly active, and they signal all the time, essentially. All right. So after that, what we are looking at, we have seen this picture all the time, that these are the class 3, that's as we have seen right here. These are class 3 intracellular transducers. And the classic example of this that we just out of the ones that we talked about is RAS. So what happens that many of these proteins essentially is a GTP bound, inactive. And when that GTP is essentially exchanged by the GDP, this is now active. So essentially, if you just remember from your signal transduction, so you have the RAS and the SOS, which we call the son of 7 less. That SOS is essentially the GEF, or the guanine nucleotide replacing factor or exchange factor. So receptor tyrosine kinase is signaling. It binds to the adapter protein GRUB2. That binds to SOS. And then SOS changes in the RAS GDP to GTP. You can just take a look. That part is not needed, but just saying that it is GTP. And then this GTP is hydrolyzed and with the help of a GAP, which is GTPase activating protein. Now, most of these G proteins, small molecules, they have intrinsic GTPase activity, but it is very, very less. It needs the help of the GTPase activating protein to hydrolyze the GTP, and it becomes inactive. Now, what happens that if you have this is what we are talking about, that if you have this glycine 12V, this glycine to valine mutation at amino acid 12, or say a Q67 to L mutation, the glutamine to leucine mutation, at that time, what happens? The GTP remain bound with the RAS constitutively. This cannot be hydrolyzed anymore. And we have seen that the similar thing, but remember that this is the G protein small molecule in the cell. This has nothing to do with the GPCR activated G protein, which are associated with the membrane. But we have seen that say, for example, the cholera toxin locks those the alpha subunit and the GTP, and it is constitutively active. And here, just a mutation makes it constitutively active because it's bound to the GTP and it's constantly signaling. And oncogenic mutation causes RAS to be constitutively on. This is one of the very, very strong oncogene mutated in many, many cancer. And these are some of the common mutations that is found in more than 50% of all cancer. So it's amazing. All right. So after that, we're looking at, so this is essentially the same picture, class 3, but earlier in class 2, we saw that the mutation was here in the receptor. Here we are looking at the receptor. It's in the intercellular transducer where the mutant RAS is activated and growth. So essentially, that's what we are seeing right here. So essentially, with the way we ask, say the previous one, let's see that this is associated with a question. So we can ask is that in what way the RAS oncogene essentially contribute to the cancer? And just remember that RAS is essentially a GTPase. It's a molecular GTPase. And this is mutated, and the mutated form cannot be switched off. Remember that it's a GTPase, and the RAS, which is right here, activated RAS, and it cannot be switched off, keeping the pathway on. So this is exactly the way we can ask a question from that. This will do it. All right. All right. So sometimes we actually ask a question from here like this. So this is actually a question. So very carefully know the question, because this question, there is a tendency a lot of people actually miss this question. Sometimes we just give you a statement like this, that which of the following type of mutation cannot give rise to an oncogene? And we give you some of the scenarios, which are the ones that we have seen. And I'm just going one at a time. Suppose we are just giving you an example, a point mutation changing just one amino acid in the protein product. See, valine to glutamine, or G2V mutation, Q2L mutation. These are all single mutations. So this single point mutation can give rise to an oncoprotein or oncogene, essentially. On the other hand, what happens with the other one that we can say a translocation, which puts the gene under control of a strong promoter, producing an oncoprotein. We are going to see that in a second in this one. And then we also have the translocation result in the formation of a fusion or a chimeric protein like this. So we can just have this chimeric fusion protein, then single point mutation, and promoter under a strong promoter control. These are all. But if we have an example where we say addition or deletion of a base producing a nonsense message and an inactive protein product, if we have that cannot give rise to, that is not an oncogene, cannot give rise to an oncoprotein. Because what you mean by a nonsense message is essentially you introduced a base, and then the entire reading frame of the protein is actually thrown off. So it produces a protein which has no function. So it will be produced and immediately it will be degraded with a degradation pathway. A nonsense protein which doesn't have any function cannot be an oncogene or oncoprotein. It will be just a mutated protein that has no function, but it cannot be. Oncoproteins must have to activate a pathway or suppress the ability to do something. So essentially, that's say for example, if you just can think, okay, all right. So if what happens that if you have a p53, which essentially we're going to see that if it is supposed to check the DNA repair, and if you have a nonsense mutation, it is not producing any p53 at that time. Essentially, what happens? But essentially, what we said that p53 is not an oncogene; it's a tumor suppressor gene. So remember that tumor suppressor gene is an entire story. So what we are asking that remember that is actually important, because you have to remember from all the way that the proto-oncogenes are normally off and they needs to be on. So it has to be a protein which is mutated and on. Tumor suppressor genes are essentially all the time off, and you turn them all the time on. I'm sorry, all the time on, you turn them off. So essentially, if you have a protein which is produced all the time and you just turn them off, that will throw it off essentially. So just remember that how it works essentially. So just remember that example, that inactive protein product, nonsense protein will not going to lead to those if we just say it in terms of an oncogene, but not tumor suppressor gene. In tumor suppressor gene, yes. If you have an inactive p53, that can lead to the cancer. So oncogene or tumor suppressor gene, that you distinguish for that type of question. All right. So then you have a class 4. This is a transcription factor. So if you just go back all the way right here, class 4, last set of proto-oncogenes. So a transcription factor. Here you have the chromosome 8 and 14. And what you have that you have the MYC, which is a positive regulator of cell cycle. It's on chromosome 8. And then on chromosome 14, you have the part which make antibodies. So this is a variable heavy chain coding region. This is a constant heavy chain produced region. And here is a picture of an antibody essentially. And what you see that these VL, variable light, these green parts are essentially what we call as a complementary determining region, or CDR. This binds to an epitope or a structure that it finds and fit properly. Whereas this variable heavy VH, these variable heavy chains are right here, which also helps in the binding of the antigens as well. This is light and this is heavy, but the constant heavy is same in one isotype of the immunoglobulin. So say for an example, I don't have to go detail, but the most common one is IgG, immunoglobulin G. So in immunoglobulin G, this constant region in all IgG is the same. What it varies the billion times and stuff is these two regions, variable heavy and variable light. So these are producing the constant H and the variable heavy. And there are some ways that they just do a lot of these rearrangement to make this lot of these different changes for the antibodies. But that's an entirely different ball game, not needed. But what happens that here you have a chromosome break, and then you have a translocation between the chromosomes. So right here, the MYC actually breaks from here and is translocated to the chromosome 14. And we call this, the arms are, the top one is the P and this is Q. So here you have a Q plus, whereas Q minus means that you have lost a part of the Q arm, and instead of the MYC is here and the VH is transported right here, variable heavy. So essentially, this is a Burkitt's lymphoma. So essentially what happens that this is a leukocyte, or this is essentially a cancer of the B cells, which produce antibodies. So there's a problem with the antibody. You have a variable heavy region here and a constant heavy region here. And the variable heavy region actually doesn't produce too much of the antibodies, and they will be to some extent immunocompromised. Their quality of the antibodies will be less, but that's a different story. But the more drastic effect is that both of the CH and the VH, they are under a control of a very strong promoter, because the B cells are constantly producing these antibodies. So these CH and VH are producing their proteins constantly. Now, instead of VH, you have the MYC. MYC is always under an extremely tightly regulated promoter. That means it is constitutively always off. Only when you get a signal, it turns off. Now it is under a promoter which is on all the time. So this is the promoter, and the MYC is expressed all the time. So the translocation acquired immunoglobulin gene control region has a constitutively active promoter enhancer. The gene is always on, driving transcription, resulting in uncontrolled cell division. So transcription will produce the proteins required for the cell cycle to move, and this is uncontrolled cell growth. So that, again, that is a type of question that we actually asked, is like which of the following type of mutation cannot give rise to an oncogene? So if we just actually say that a translocation that result in a formation of translocation which puts the gene under a control of a strong promoter producing overexpression of an oncogene, this is also a way that an oncogene can produce that. But again, I'm just repeating that last one, that which is not, is that if it is an oncogene, and it is only for the oncogene, not for the tumor suppressor gene, right here, for the oncogenes, that if there is an addition or deletion of the base producing a nonsense message, and if it produces an inactive protein out of these 1, 2, 3, 4, that cannot give rise to the cancer, because protein has to be active. All right. So should we take a break, or should we finish it? What do you guys think? Let's go on. Is it you guys good? Okay. And am I still okay with the connection wise? Not breaking up? Okay. Okay. Perfect. So yeah, I mean, like how many chapters we have? We have but this part again will not require. So this will not take much time. So right here you have a tumor suppressor gene, and the RB is the first tumor gene identified. Normal inhibition of cell growth proliferation, and it's a recessive gene. Again, as I said, that is that one detected copy does not cause tumor. You have to have both as small a's, capital A, or capital A, small a. That does not matter, because this is on all the time. But it is inherited in a dominant manner. And this is where all of the people actually get thrown off, that it's a recessive gene, but it's inherited in a dominant manner. Means that if both copies are defective, to show the dominance expression of the tumor. So I'm just explaining that what it means. So you can have three conditions essentially. AA, remember that. So AA right here is not producing protein. Remember that from the previous one. And AA, it is producing protein. So the small a is good. And A right here is essentially producing the protein. So remember that. So again, if I just, so I'm saying protein right here is a protein, no. And small a, protein, yes. So small a is producing protein and the capital A is not. So essentially, why it's essentially we calling is a recessive gene, because if this is the condition, see that right here, you have a small a, this is producing the protein. So in heterozygous condition, if one allele A is correct, it's not going to cause cancer. So recessive, because you have to have both of the allele homozygous for not producing the protein. So essentially what happens, if you suppose you have like a dad like AA and you have mom like AA, at that time, whatever combination you actually get, you are never going to get a small a in all of them. Just do your basic basic genetics, that if you just have these, so you can only from this, you can actually produce AA. So on the other hand, if you just take a look at that if your dad is like this, AA, and mom is RB defective, so at that time, so you have an AA from here, or you have an AA from here and here. You cannot get both the alleles from the mom. So here, this is also not going to do it. But if, say, for an example, if his dad is like this, and then mom is either like this and either like this, then you have a chance to get an AA. So that's why we say that it is inherited only in a dominant manner. And to show the dominance, because it's a recessive gene, both has to be in a homozygous recessive condition. And that is what is the meaning of that. So it is not an oxymoron. A lot of people actually say it's like it's a recessive gene, but what do you mean by in dominance manner? Is that the only way that you can get, if you get a small a effective, where the protein is a, so I actually completely said it the different way. Hold on. I said it in a different way. So I wrote it down that protein not produced and protein produced, so it will throw you off. So let me take it back in here. So hold on 1 second. I'm so sorry. So because we keep it that way, that A is not producing protein and small a is producing a protein from the proto-oncogenes the same way. That is. So remember that the tumor suppressor gene is always on. So that's basically what it is. So it is always producing AAs. So let's go back to AA. So here you can see that if your mom and dad is AA, protein is produced. Absolutely no way you can get a protein. So if you have a condition AA, somebody has an AA, you cannot get. If you have an AA as well as AA right here, there is a possibility you can get that. Am I saying it correctly? No, not saying correctly. Again, doing the same mistake. So see that right here. I'm so sorry. I'm sorry for confusion. So this AA is producing constantly the protein. So now what you have, that if you have a condition where you have AA and here you have mom as AA, so one of the allele is not producing protein. And this is producing protein. So none of them have the problem. But if they produce this, there is no protein synthesis. So if from here, if they only become AA, which is dominant, at that time, protein will not be produced, and that will only cause the cancer. If it is capital A, or small a, these are all the conditions where you are all good. You are producing protein. So only for the disease to happen, capital A allele, which is not producing protein, that needs to be inherited, which is what we call as a dominance. Does this make sense now? Or should I repeat it? Are you guys good? I just wanted to make sure because I confused that first. Yeah. So just remember that as soon as you see that, so all you from here, you have to remember that for your tumor suppressor gene, they are constitutively on. That means that they have to be producing the protein all the time. And here we mentioned that small a means that protein is produced all the time. Capital A mutation means protein is not produced all the time, and this is off. If this happens in both the alleles, that will lead to essentially tumor suppression. And as it says, as a both allele, which is this, we call is as a dominant gene, but it's a recessive gene because what happens that it is essentially is constantly on. That's essentially the character. So that's essentially, and that's why the defective copy does not cause tumor. So homozygous dominant. So no, it is just that letters does not mean anything. It's empirical. So homozygous dominant is bad for a gene where I designate the capital homozygous dominant is not producing the protein. So this is just like totally empirical, like the way I can say that this is probably a better idea. So think about that, RB plus and RB plus. That means this is producing the gene. Let's convert that to that. And then you have an RB plus and RB minus. This is also producing this. So this is good. This is good. Only if you have an RB minus and RB minus of the allele, this means that this is off, and this can essentially lead to the cancer. And this RB minus is essentially to show dominance. Essentially, it's a recessive gene. That means that essentially what happens that if there is only one allele functional, the disease doesn't express. If both, for the disease to be expressed, both the copies must be defective. So it must be received in a dominant manner. That means you have to receive a wrong copy from both the parents. Does it make sense? Good. Yeah. So I mean, like, if we just write it down to the RB plus, RB minus, I think that instead of A or small a, that's much easier. Yeah. In other words, you need 2 copies of tumor suppressor mutation to mutations to lead. The dominant is that even though if you have a monitored mutation, the normal copy can recover. Yes. So if you see that is correct. Yes. So if you have just one copy that is functional, no cancer. You have both must be mutated and bad, which is non-functional. Yes, that is correct. Make sense, Stephanie? Good? Okay. All right. Yeah. So I mean, like, if you just get that RB plus, that's what it is. But again, we are not going to like the wordings are like that. But essentially, the typical type of questions that what we ask is, say, for example, familial retinoblastoma is inherited. So if we just say that familial retinoblastoma is inherited is in a dominant manner, while the mutation of the responsible for the gene are recessive, and they are tumor suppressor genes. Remember that. So the genes, it is received is a dominant manner, but the gene responsible for cancer are tumor suppressor genes, and they are received and they are recessive genes. So again, I'm repeating. Sometimes we ask, is that which of the following is a correct statement? And we say that the familial retinoblastoma is inherited in a dominant manner. It's a tumor suppressor gene, and it's a recessive gene. So the gene is recessive, but inherited in dominant manner. If you just remember these 2 words, the gene is recessive, inherited in dominant manner, you're good. So these are the 2 words that you just write down. And this will essentially, if you just look it up, you should have no problem with those. All right. So let's see. So essentially, sometimes we give you a kind of a like an idea. We can give you some graphs sometimes. So let's take a look at this. So right here, so we're spending a good amount of time. And suppose there are something like this, and after that we are looking at this. And suppose we are just giving some of the names of these things that what they are producing. And we say that this is an L8 minus and L8 minus. And we are saying, let me take this one off, like right here. And this will, otherwise it will confuse. On the other hand, we can see right here and right here. And here we have a condition where you have an 8 plus and L8 minus, and we have an L8 minus and L8 minus. And suppose this is essentially, if you have this L8 location, you have the mutation. Both of them are minus, means that they do not produce the protein. L8 plus and minus, that means L8 plus is producing the protein. And you have L8 plus and L8 plus, both the alleles are producing the protein. And here on the Y, this is the occurrence of cancer so you have the patients look at their gene composition, and you see that if a patient have an L8 minus and minus, the occurrence of the cancer in them is the highest. If you have 8 plus or minus, your occurrence of them is much less. If you have L8 plus or 8 plus, occurrence is much less. So based on that graph, you actually predict that what kind of gene it should be. So you can easily see that it's L8 minus or L8 minus. So this must be a tumor suppressor gene. When both of them are defective, that is essentially growing the cancer. Make sense? Yeah. On the other hand, if you just think about this way, that if we just go back, and because this is, I'm spending a good amount of time on this one because that this will actually help him here for the proto-oncogene. On the proto-oncogene, that graph will be entirely different. We can easily see right here, you have this, and then you have this, and you have this. So in here, I have to do that. If you just do the L8 thing, essentially, what we have to do that for here, you have to have right here, suppose this is L8 plus, and this is L8 plus, and then you have L8 plus and L8 minus, and this one is L8 minus and L8 minus. So plus means producing protein, minus means not producing protein from the allele. See that when both of the allele are producing protein, the number of the cancer is much higher. If one of them producing the protein, still cancer is higher. But if they are not producing the protein, which is essentially the turned off gene, you have much less cancer. And this, if you give this graph and tell you that which it is, this will be a proto-oncogene. Make sense in between the two? Good? Okay. All right. So let's see. Let's move on. All right. So here, these are just a very straightforward process that how you have the these tumor suppressor genes are acquired, which are essentially the double mutants. Essentially, this is a missegregation of the chromosome. Very straightforward. One copy, one chromosome gets just only one copy of the chromosome, and it dies. Another gets a trisomy. It gets rid of the wrong pair, and if it does have those. So right here is a plus and minus. So no problem, heterozygous for mutant allele, but this is a homozygous and it received it in a dominant manner. This is your dominant manner, and this will essentially express, not express the protein because it's a zero means like a no protein, and that will lead to cancer. So this is one of the type we just can ask, what are the different ways that you can get the tumor suppressor gene, loss of heterozygosity, a missegregation. Another is a mitotic recombination. So mitotic recombination between the homologous chromosome actually happens. This is a normal phenomenon. This is not meiotic. This is not crossing over. This is not meiosis. This is double strand break in chromosome and exchange of the part with the homologous chromosome. This happens during the mitosis, discovered like almost 90 years ago in Drosophila. So essentially, if there are wrong copies and two of them are defective, you can have a mitotic recombinant homozygous for the mutant allele. So this is another possibility essentially. So we can just ask, is that what are the ways that loss of heterozygosity can be achieved? Can occur as a result of chromosome missegregation, can occur as a result of recombination between the homologous chromosome. These are the two ways that this can happen. Just very straightforward. All right. So after that, we are looking at a couple of cell cycle control proteins. So here you have a cyclin D CDK4. We talked about that. and the cyclin D CDK4, after it produces, there is a protein called P16. It binds to this, and it's an inhibitor. And when this P16 actually binds, it doesn't allow the cyclin CDK4 to activate it. When the P16 is removed, essentially, cyclin CDK4 is active. That will phosphorylate the RB, which is bound to an E2F, and this is a transcription factor for the cell division, essentially. So when E2F is released, that is activated for transcription of protein needed for the DNA synthesis. It will go to the S phase. So the CDK4 essentially phosphorylates the RB. If the RB is phosphorylated, it lets the E2F free, and that will lead to cell division. So that's the process. But what happens? There are two tumor suppressor protein here. If you have a loss of P16, cyclin D CDK4 is constitutively active. So it's constantly going to phosphorylate RB, and it's constantly going to make the E2F free for DNA synthesis. On the other hand, loss of RB, we already talked about that. Loss of RB means that the RB actually, so loss of RB sometimes means like where the connection. Loss of RB essentially means that it doesn't have the ability to bind to E2F. It cannot sequester E2F after it is produced. So the E2F, when produced, is free all the time because RB has lost its affinity to bind to E2F and sequester it. So it cannot control E2F anymore. E2F is constantly active. So loss of P16, RB, both of them are tumor suppressor. All right. So here you can see that right here. Again, you do not need to go over the cell cycle. We're not teaching cell cycle here, but we're just showing you that where the P53 works, and it's a role in a DNA damage checkpoint throughout the cell cycle. So you can see that there are 4 DNA damage checkpoints. You do not have to remember those, but essentially the way it works, that this ATM or ATR, these are ataxia-related proteins. These ATM and ATR, 2 different proteins. One is called ATM, another is called ATR. They actually scan the DNA and look for mutations, unreplicated DNA, or DNA damage. If they actually see that there is a DNA damage, they actually stabilize P53. And P53 is a transcription factor constantly produced in the cell, and it get destroyed. Destroyed means like it is degraded very quickly. Half-life is 30 minutes in a normal cell. 30 minutes is turnover is 30 minutes. If ATM and ATR, these are kinases, if they phosphorylate P53, it is not degraded anymore and it's stabilized. Now it binds to a transcription factor which produces a P21. CYP is a cyclin inhibitory protein. So P21, just like the P16 right here, just like the P16, P21 is a 21 kilodalton protein. It binds to the cyclins essentially, and when it binds to the cyclin, it makes it inactivated. So the corresponding CDKs, which are the proteins needed for the phosphorylation of the substrate to make them active, they are inactive. So this is essentially the checkpoints by the P53, DNA damage control checkpoints. So how this essentially works is, again, P53 stabilized by the DNA damage, induce synthesis of P21 CYP, bind to the cyclin, arrest of G1, G2, essentially. And P53 transcription factor regulates the cell cycle. So we just ask a typical question: which of the following statement correctly describe P53? So remember that activation of the P53 leads to the inhibition of the cyclin CDK complexes, and P53 binds to the, so that's essentially cyclin CDK complexes. That's its activation of P53. That would be a correct answer. All right. So here is a little bit more detail, and that's pretty much, this is the last slide for, after that is just a couple of information. So what happens, the damaged DNA is sensed by the ATM. It's a kinase as well, and it can activate another protein called CHEK2. Once it is phosphorylated, CHEK2 phosphorylate this CDC25, and it actually causes the proteasomal degradation. This is a rapid G1 block. So where is CHEK2? If you can see that right here, this is CHEK1-2. Right here, this is CHEK1-2, and this is CDC25. So both of these arms, it's a 2-prong arm. These are essentially activate the checks and also control the cell cycle. But the main thing here is that what happens that the P53 is produced in the cell. So I just jumped from here to here. P53 in normal condition, constantly produced in the cell. It binds to a protein called MDM2, and this leads to the proteasomal degradation. It's just like constantly produced, degraded, binding to MDM2, degraded, binding, degraded. Now what happens that if there is a DNA damage, technically, the ATM binds to the DNA and then phosphorylates the P53. Phosphorylated P53 is stabilized. And then it is a transcription factor. It goes to the nucleus, translocates to the nucleus, phosphorylated P53. And first it tries to do the DNA repair in sustain G1, G2 arrest, essentially. So it will induce synthesis of proteins, which are enzymes required for DNA repair. It will block the G1, G2 by inhibiting the cyclin CDKs by the CYP. And also, if it cannot essentially repair the DNA, it will also start producing apoptotic proteins for the programmed cell death. It will kill the cell if it cannot repair the DNA. So this is a normal process. Now what happens here, that if MDM2 right here, MDM2 is phosphorylated, essentially, then MDM2 cannot bind to P53 anymore. So the pool of P53 protein increases, it's phosphorylated, and do this. So this is essentially the process, MDM2. And I will come to this hyperphosphorylated in tumor cells. So pool of P53 increases. On the other hand, what happens is the MDM, so if there is an oncogenic signaling that the cell suspects that there is a DNA damage and there is uncontrolled cell division is going on, it produces a protein called P14ARF. And the P14ARF binds to MDM2, and no longer MDM2 cannot lead to the degradation of P53. It also increases the pool of P53. So as pool of P53 increases in the cell, it is stabilized, transcription activator, and everything goes on. But what happens that if you have a P14ARF, which is not produced, it will still allow MDM2 to be active, and it will bind to P53 and degrade it, and it will not increase the pool of TMS synthesis. So a mutated P14ARF tumor suppressor gene can't bind to the MDM in an oncogenic signaling, DNA damage and control cell division, binds to P53 and degrade them. So this is a typical question that we ask is about essentially that, say, for example, if we just see that we say it like this, that a cell, say, for example, suffered from a mutation in the RAS oncogene, leading to a loss of control of cell division. This event essentially induced a protein called P14ARF, like could be any stories, and which binds to the MDM2 and renders it inactive. So no longer it can bind P53. And then we ask that what type of protein is P14ARF? Remember that it's a tumor suppressor protein. If it is mutated, then it has not binding to the P53. P53 cannot essentially suppress tumor. That's essentially the process. Now, the question where everybody gets confused, and I get a lot of email, I'm going to post a discussion as well. Several of you asked this question, that why we see that in a cancer cell, this MDM2 is hyperphosphorylated, and you have a higher level of P53. We actually see this in many cancer cells. So in diagnostics, P53 imaging or P53 staining of a biopsy or a cancerous tissue is a very common. So you stain with an anti-P53 antibody, and if you look that the P53 level is more than higher in a normal cell, that is most probably a cancer cell. If you see too much hyperphosphorylation, that MDM2 is probably a cancer cell. So the question is like in a tumor cell, this is the increase P53 means it is doing DNA repair, sustain G1, G2 arrest, apoptosis. The problem is that the P53 is not mutated out of those 3-7. There are, out of those 3-7 mutation, some other oncogenic proteins have been mutated, and P53 is trying very hard to control the cell division by increasing its amount. it is hyperphosphorylating, ATM is hyperphosphorylating NDM2 to increase the pool of protein synthesis. It is trying hard, but still it is unable to do so. Does it make sense? So essentially what I'm saying, that in a normal cell, P53 is very less, but in a because there is no damage, but in a cancer cell, the P53 amount increases all the time. So you will suspect that higher P53 means that it is trying to repair the DNA and it should be fine. But again, as I said, that P53 is doing its function, or the damages are somewhere else and is so severe that P53 somehow got mutated, it is getting phosphorylated and cannot. Suppose, say for example, some of the apoptotic proteins, they are expressed without P50, they are not expressed even in presence with the P53. They cannot control some of the G1, G2 arrest because RB is active all the time. P16, we say oncogene, that is essentially a tumor suppressor gene that is defective. So the mutations are somewhere else. And this P53 is trying its hard way to do this. And the same way MDM2 also increases because it's trying hard to control the cancer, but it can't. So this is a typical explanation of the process. All right, so you can see that right here, P53 greater than, so let's go back, okay. So MDM, so what happens is that the MDM2 essentially may not be even mutations. So essentially what happens that MDM2 is being hyperphosphorylated because the cell is sensing that there is damaged DNA or uncontrolled cell division going on. So it is inducing MDM2 to increase P53. But even despite of higher amount of MDM2, which will not degrade P53, or even despite higher amount of P53, the cell is still unable to control that cell division because there are other proteins somewhere else. That's why we said 3 to 7. MDM may be perfectly normal, and it is hyperphosphorylated in somewhere. See that MDM is a tumor suppressor protein, so it is produced all the time. And essentially what happens that if the MDM2 is normally inactive, it will just increase the P53. But that does not mean that that cell is essentially damaged in that process, because if it's a normal P53, it will induce apoptosis and kill the cell, and it will not tolerate. But that is not cancer. Here, MDM2 is perfectly fine. It is phosphorylated. It is trying to increase the pool of P53. P53 is trying to do that, but right here in this pathway, in this transcription, even without the help of P53, these genes, think about this way, that these DNA repair proteins are essentially synthesized by the P53 transcription factor. And the DNA is damaged in the transcription, damaged in a site where P53 actually binds. So the binding of the P53, suppose, is not happening despite P53 is active. So that means that it will not be able to synthesize DNA repair enzymes. It will not be able to make the cyclin inhibitory protein for the G1/E2 arrest. It will not be able to produce the apoptosis. So the DNA can be damaged, P53 may not be damaged, and that will abolish its ability to bind to the DNA site to control the transcription. So the transcription, even is an active P53, perfectly normal, cannot drive the transcription because the binding site is constitutively active and it is activated by other transcription factor. So that's essentially the process. Yeah. Okay. All right. So here it's the 70% of cancer. RAS and P53 are some of the thing. And see that this is where the most important part is, that if you look at these regions 1, 2, 3, 4, 5, you have hypermutations. And if you look at these, out of those, R, these are all arginine sites. And can you tell that why those are in there? You can think that why those are all arginine mutation. There's one glycine, but most of them arginine. Think about that, what is that does. RDKs? No, not RDKs. Arginine is, so this I'm giving you a hint. Arginine is a positively charged amino acid. And think about that, what P53 does. So ATM, ATR, DNA is negatively charged. These are the sites where it binds to the DNA and get phosphorylated by the ATM, ATRs. So these are plus charges, and these are the DNA binding grooves of the P53, which is essentially binds, locks onto those. So basically, the ATMs actually sit near the damaged DNA, and P53 goes and binds to the DNA, and it gets phosphorylated by the ATM. So the ATMs are like sliding mechanism. It slides around with that. And when P53 binds to the DNA, it finds P53 and phosphorylates them. So these are the DNA binding domains. That's it. So plus charges binds to the negative charge because the phosphate backbone of the DNA is negatively charged. That's why. And here, this is probably the last question, and very straightforward from here is let's right here. And essentially, we ask just very straightforward that what are some people say it's a BRCA1, BRCA2. Some of the oncologists actually pronounce them as a BRCA1, BRCA2. So everything is correct, I guess. So these are implied in hereditary breast cancer, and these proteins are DNA. So this is a class 6. These are the DNA damage repair proteins. So essentially, the way is that P53 induce the synthesis of this BRCA1, BRCA2. And suppose P53 is even inducing BRCA2, but there is a DNA break in here, which is the chromosome 13, and it cannot damage the DNA. So essentially, what is happening in here, P53 is trying to induce or make BRCA1 and 2, and that gene is already mutated. It is not producing any protein. So it cannot, and the loss of chromosome genome integrity. So P53 has nothing to do with it. It is just increasing its amount, but its effort to make BRCA1, 2 is not possible because it is mutated as well. Yeah. ATM, ATR are mutated. It can be mutated. Absolutely. Yeah, those are ATM, ATRs are absolutely tumor suppressor protein. Yes, absolutely. And it may not sense the DNA damage. Yes, these are all of these proteins in this pathway. Their ability is to check everything, and anything that it wants to check or stop is a tumor suppressor protein. And if so, ATM is produced all the time for this checking, and ATR not produced, it will essentially cause cancer. Yeah, absolutely, it's implicated. Yep. So and loss of chromosome, unregulated transcription, unregulated protein ubiquitination. So which is essentially, so any protein added with an ubiquitin is essentially going to get degraded, and loss of cell cycle control. Yeah. So that's pretty much will do it. That last part is essentially mainly for some of the areas that the works are. And, even in the school, you see a lot of different type of interest in the students. They want to ask some of the question, and these are some of the things that what are the things that are available that what we are doing in here. So if you're just interested, that you just can go on each one of these, and just this is just like a guideline that where you can look at some interesting literature that they are working on those, and that's why I just put those in there, but this is not required. All right, so that's it pretty much.